Let's see the mechanism of enzyme action, catalysis. An enzyme is a three-dimensional globular protein that has specific chemical composition due to its component amino acids and a specific shape. Every enzyme, by virtue of its specificity, recognizes and reacts with a special chemical substance called substrate. Any enzyme therefore reacts only with its specific substrate and transforms it into products. It is then released unaltered and thus can be used again and again. Enzyme plus substrate forms enzyme substrate complex, which forms enzyme plus product. In certain cases, enzymes act in a series of chemical reactions in a particular order to complete a metabolic pathway, such as respiration or photosynthesis. The successive enzymes containing these reactions are normally present together in a precise order of reaction, such that substrate molecules can be literally handed on from one enzyme to another, forming an enzyme to enzyme chain. In this way, the products from one step in pathway are transferred to the enzyme catalyzing the next step. An enzyme and its substrate react with each other through a definite charge-bearing site of an enzyme called the active site. The charge and the shape of the active site is formed by some amino acids present in the polypeptide chain of the active site of the enzyme. These amino acids are brought closer and are arranged in a specific way by coiling and folding of the polypeptide chain within the globular symmetry of the enzyme. The active site of the enzyme is made up of two definite regions, the binding site and the catalytic site. The binding site helps the enzyme in recognition and binding of a proper substrate to produce an ES complex. This reaction activates the catalytic site. Activated catalytic site catalyzes the transformation of the substrate into products. Thus, the enzyme, after catalysis, detaches itself from the products unchanged. Enzyme require aqueous medium for its activity. Emil Fischer in 1890 produced a lock and key model to visualize substrate and enzyme interaction. According to this model, as one specific key can only open a specific lock, in the same manner a specific enzyme can transform only one substrate into products. According to lock and key model, the active site is a rigid structure. There is no modification or flexibility in the active site before, during, or after the enzyme action, and it is used only as a template. Later studies did not support this model in all reactions. Most enzymes do not float about in a kind of cytoplasmic soup, but are attached to the membrane systems inside the cell in a specific and orderly arrangements. Mitochondria and chloroplast are good examples of this. On the basis of the evidences, Koshland in 1959 proposed its modified form known as an induced fit model. He argued that when a substrate combines with an enzyme, it induces changes in the enzyme structure. Change in the structure enables the enzyme to perform its catalytic activity more effectively.